Welcome to this biochemistry lecture on pathways of catabolism, and this is a general overview. We're going to talk about all the different pathways that exist for catabolism, and from this chart you can see we're going to start at the top with carbohydrates. So we bring in carbohydrates, we'll talk about breaking them down into monosaccharides. That allows the saccharides to go into glycolysis. From glycolysis, we go through pyruvate, acetyl-CoA, into the citric acid cycle, and down through oxidative phosphorylation. We also have two other mechanisms by which we can get into. We have beta oxidation of fatty acids. That is where you see on the left-hand side with lipids. And we have proteins. Proteins can also be used as energy, and they also require an additional cycle to get rid of the amine group, and that's where you're going to see the urea cycle come in. So, and there's a few other things that we'll discuss in here, gluconeogenesis and a few other things that exist that will kind of round things out. But this is the general pathway of catabolism. There's two, and generally we talk about the workhorse, and the workhorse of this process, and the important part for you to know, is the citric acid cycle. It's also known as the Krebs cycle or the tricarboxylic acid cycle. And then we go into oxidative phosphorylation, also called the electron transport chain. So we will get into all the details on this topic. Generally speaking, metabolism is the sum of the chemical reactions that we're going to use to break down biomolecules into smaller ones, and that is going to be used for energy and to build up, and it uses the products to build up more specific biomolecules for the organism. So, if we take in plants, they have a specific set of biomolecules they use. We break those down, and we'll use those parts, those components, monomers, into on the other side, which is anabolism, and that allows us to build things back up. And we also are going to have an element where we're going to excrete things out. So that kind of, hopefully, and that's what we're going to talk about for the next 2 weeks. This week we'll talk about catabolism. Next week we'll talk about anabolism, and the next two sessions, or a little bit of units, I guess I should say. We'll talk about how we get to break things down and what energy comes out, and then after that we'll talk about how do we build things back up from their subunits. This also kind of gives you an idea of what's coming in and what's coming out. So we have small, carbon units coming in. It goes into the citric acid cycle. We're going to generate NADH and FADH2. Some of our byproducts are carbon dioxide. Once we get these NADH, FADH products, and also protons, we are going to then, across mitochondrial matrices, we're going to generate energy from that, which is ATP, which is what you see at the bottom. We'll pump out electrons out, and we'll generate and these carriers, the NADH and FADH plus, or FADH2, are carriers, and so they those carriers provide the ability to also generate the energy they're also part of the process where we're going to generate energy. So there are 4 general compounds that we're going to see: ATP, FAD plus, NAD plus, and coenzyme A. We're going to see these all the way through the metabolic, in particular the catabolic pathways. And so you will see ATP, AMP, and ADP are the agents that allow us to store energy, and through the phosphate groups. So through this phosphate linkage, through these high energy phosphate bonds that we have, these phosphorus bonds provide a significant amount of energy that gets transferred through coupling reactions to enzymes that allow the entire work of the cell to be completed. I'll keep re-emphasizing that throughout this whole thing. That's the general gist of that molecule. This is the ultimate goal of all of the processes that we're talking about. Let's generate ATP. We have FAD, which is a carrier of electrons. So we transfer electrons through there, and through oxidative phosphorylation, through oxidative reduction reactions, I apologize, through oxidation and reduction reactions, we will transfer these electrons back and forth, and those electrons allow us to generate ATP. NAD plus is also in this. It does also the very similar thing. It transfers through oxidation of NADH to NAD plus. We generate 2 electrons and 2 hydrogen, and again, those are electron and hydrogen. They're mainly electron carriers. They also generate hydrogen. Those hydrogen can be used to generate ATP, as can the electrons. And last but not least, we have CoA. It's coenzyme A. It's abbreviated CoA or CoA-SSH, and it's the agent that allows us to transfer an acyl group throughout the process. You can see it's pretty complex. It's got a whole bunch of different functional groups that are associated on that background. You have sulfur, phosphorus, benzene groups. You can go on and on what's on that molecule. It's one of the more interesting molecules, but it's the acetyl group, and we'll talk about that in detail as we get going. But these are the molecules that are common throughout the pathway. So the citric acid cycle is going to be one of the workhorse processes in this whole catabolic pathway. Again, the ultimate goal of this whole pathway is to generate ATP. You can see that we see all of these molecules we just discussed in some form or another. We have acetyl-CoA as the introductory step. From there, we're going to transfer that CoA off. We're generating NADH and a proton in the second step of the citric acid cycle. In the third step, we generate nucleotide energy. In the fourth step and the fifth step, we're using FADH. In the fifth step, the last step, we have NAD plus, or NADH and proton that's generated. So all the way through this process, we're generating these molecules, and they'll become an important element in carrying into oxidative phosphorylation with the ultimate goal of generating ATP and thus energy. This is a very simplified view of this whole process. 2 things that are probably important in this. Note that along the steps we're generating these molecules to generate energy. This is just the product, or the reactant and the product. For the energy side of things, there's obviously a lot more that goes into each step, which we'll go in greater detail. Also note that there are waste products, right? CO2, that are also generated. They're in blue in this cycle. We'll dive deep on citric acid cycle in an entire lecture and go over all the steps, the enzymes, the cofactor, and the regulation of all this process as we get going. But just note that there are molecules that come out of each step of this process that are important in generating energy. So the citric acid cycle, tricarboxylic acid, the Krebs cycle, all the same thing. Again, starts with proteins, carbohydrates, fatty acids. We go through glycolysis into pyruvate, into acetyl-CoA. We have intermediates of the citric acid cycle, and those intermediates then spin off as we go from intermediate to intermediate, spin off molecules that allow us to generate the energy in oxidative phosphorylation. And but the important part of this is you can see that we feed into this cycle over, just give it a different view of feeding in these biomolecules in this cycle in order to generate energy. From there, we go into oxidative phosphorylation, where we have 5 workhorses, complex 1, 2, 3, 4, and the FOF1 ATPase. And some people lump the FOF1 ATPase in the complex 4. We'll just refer to it as the FOF1 ATPase. Again, some people reference it as complex 5, but again, we'll just talk about it in terms of FOF1 ATPase. It's a really fascinating molecule, as it's a motor. It's a pump and a motor. It rivals anything that you'll find that we have manufactured. In fact, it's probably better. Anyway, different story. We talk about flow of protons, flow of electrons. Your NADHs are electron carriers. Your FADHs FADH2 are electron carriers, and what we're doing here is we're moving molecules to generate energy. And each step along this process, and we'll go into great detail, but what basically oxidative phosphorylation allows us to do is at the end of the day, we're pumping molecules across to generate ATP. The ultimate goal out of this is to generate ATP. And that, if you get anything out of this lecture, get this, that we take all these molecules and we're putting these things through the process. And the important thing that we get out is ATP, because ATP allows our enzymes to function. It's the energy that gives enzymes the ability to do their job. And we all know what enzymes do, the work of the cell. And so this provides the energy to do the work of the cell. And we'll go over again in great detail all of this in later discussions. One thing I just want to point out, while a lot of this work occurs in the cytosol, like glycolysis, most of the steps in glycolysis occur in the cytosol, and most of the steps in the TCA cycle happen in the cytosol. A lot of the cells of a lot of the work, and actually the mitochondrion was designed to, or mitochondrion in general, were designed to, that's their sole purpose, is to generate ATP. So we take oxidative phosphorylation, happens on the inner membrane of the mitochondria. And so that's the important part of that location, is where most of the work, it's an organelle, it's the place where the only purpose of it is to make ATP. So it generates, it's the powerhouse. So we'll talk about what the inner membrane means, the matrix means, all that kind of stuff. Krista means, all that good stuff in the discussion when we talk about oxidative phosphorylation. This is the important part of what we have. At the end of the day, the goal is to generate energy. And you can see we take in glucose and oxygen, very important component. We'll talk about this again as well. And we generate 6 carbon dioxide, heat, and water, and the heat comes out as ATP. So we get a delta G of negative 2,800, almost 2,900 kilojoules per mole of glucose. So we're getting out a significant amount of change in energy. Again, that allows enzymes to knock off that activation energy. We're transferring energy, we're coupling reactions. So enzymes group things together, and then the ATP provides the energy necessary to overcome the activation barrier and allow a reaction to occur. So that's the importance of this process, of these processes. So we take all these disparate biomolecules, and what we're going to talk about for the next little bit is how do we use it to generate energy. That's the gist of the whole unit that we're going to spend. And it's a hard unit. It's a very difficult unit. There's a lot of reactions. There's a lot of memorization that occurs. And there's not much you can do except for just straight, plain, bang into it and memorize. All right. Well, we're going to dive in. Thanks for listening.